Assalamu alaikum. This is the tutor from Ilm Online Academy, and I'll be teaching you the part 2 of paper June 2021, May June 2021, IGCSE Biology. So let's get started. Okay, so question number 4 says that two identical potted plants were used to investigate plant responses. Plant A was placed on a clinostat that continually rotated and clinostat that continually rotated. So this is the clinostat which I have mentioned right here, right now. Sorry, just a minute. Okay, so this is the clinostat right here. Okay, I hope you get it. Okay, that continually rotated. Plant P was not rotated, and both plants were then placed on their sides and kept in the dark. Okay, now figure 4.1 shows the 2 points at the start of the experiment and after 7 days. So this is the one which is showing at the start. You can see both of the plants are being shown at the start and they are pretty similar at the start of the experiment. And after 7 days, what we see is that plant A is all straight, right? And plant B, the stem of the plant B, or the you can see that it has the shoot has actually turned upward of the plant B, right? So that's that. Now state the name of the response which is shown by the shoot of the plant B. Now that is named as, now this turning of the shoot of plant B is due to a phenomena named as gravitropism. Now, gravitropism is a really important phenomena for the seedlings, for the plants, because it is the phenomena which actually ensures that the plant actually, the shoots of the plants are going to go upwards and the roots are going to go downwards, right, as they grow. ठीक है. So gravitropism, by like terms की बात करें, so gravitropism, gravi means gravity for sure, or the tropism means turning. So it is actually the growth of the plant organs or change in the direction of growth in response to the gravity. So the phenomena is gravitropism. Then explain the reason for constantly rotating plant A. Acha, so above they have told us that the plant A is actually attached to the clinostat. Now clinostat kya karta hai? The clinostat, what does it do? The effect of gravity on plant can actually be demonstrated using this clinostat. Or clinostat actually continuously rotates the plants throughout like 360 degrees in order to negate or eliminate the set direction for gravity, so that it is actually going to prevent the hormone auxin to accumulate on one side of the stem or the roots. And when the auxin actually accumulates at one side of the stem of the plant, so that is something what happened to the plant Bina. So okay, so plant A constantly rotates to eliminate, or you can say negate, this set direction, or negate the pull, you can say that, the pull of gravity um, on one side of the plant only, and is also being used as a control. Second thing is that it is being used as a control to compare with plant B. All right, so these are the two things that you need to take into consideration while writing the answer, and that would for sure, I hope that it would provide you with 2 marks required for this question. Then state the name of the plant hormone that causes the response of the shoot of the plant B, that is auxin. Then explain how the plant hormone causes the response of plant B. So remember that auxin kya hai? Auxin is a hormone and that is made in the tip of the plant stem and is made of you can say that जो meristematic tissues होते हैं, that actually make this plant stem for sure, and it actually, the hormone promotes the growth of the plant. Now, auxin hormone, what happens to it? Auxin hormone actually diffuses away from the tip, and it would get accumulated on the lower side of the stem, and that way the stem is going to turn upward. Away from the tip, gets accumulated on the lower side of stem, and thus turns upward. Turns upward. Okay. Now, seeds germinate in the soil, and the seeds that grow from seeds show the same response as shown by plant B. Now, explain the advantage of this response to the survival of seedlings and mature plants. Now, gravitropism, what does it do? It actually ensures that plant roots grow into the soil and shoots grow towards the sunlight, and it helps the plant to actually reach for for light, and light is really necessary for photosynthesis. With that, it would help plant to reach for air, and from air it would get carbon dioxide for photosynthesis and oxygen for respiration, and it would also help the plants to grow tall and for the flowers to get exposed for pollination. So these are all of the advantages. And you know that seeds, so they said the seed actually, seeds germinate in the soil and change into seedlings, and then seedlings are going to germinate into the whole plant, right? So gravitropism is actually going to ensure that roots grow into the soil and shoots grow towards the light or upwards sunlight, and helps the plants to reach for light, and light is for photosynthesis, and helps the plant to reach for air, and from air it would get carbon dioxide and oxygen. Carbon dioxide, you can mention that carbon dioxide for photosynthesis, and plants undergo respiration during the night, daytime photosynthesis and respiration during the night when light is not available. So plants also carry out respiration, and they use oxygen at night, and they use carbon dioxide during the daytime. And then plants helps ensures the plant that they grow tall, and the flowers are exposed for pollination. So you can mention at least 4 or 3 points to get three complete marks in this question, and I hope you understand. Okay, so we have got this figure, and this shows the female reproductive system, and one of these has been labeled by them, and others you have to label them. Okay, I have already labeled these, but I'll tell you the reasoning behind each and everything. So P is the site of secretion of mucus, and that has been labeled. Then Q is the site of fertilization. Now Q is the site of fertilization. Fertilization takes place in the fallopian tube, right? In the fallopian tubes, the egg and sperm are going to combine, and fertilization would take place in the fallopian tube. So we say that case site of fertilization is fallopian tube, and we have mentioned labeled it with Q. Then site of implantation. So remember that the implantation takes place in the walls of the uterus, and then next thing is site of estrogen secretion. Estrogen secretion we have mentioned here as S, and then T, site where sperms are deposited during sexual intercourse, and that is vagina, that is T. Then figure shows a section through an egg cell at the time of ovulation. Now you can see there is a nucleus, cytoplasm with stores of fat and protein, and with that a jellicoat is also present. Now explain why egg cell contains stores of fat and protein. Remember that egg cell is actually going to grow into a whole organism, right? So It actually provides nourishment to the fertilized egg, as it needs nutrients for the further division, na? For further development, for further division, for growth, for metabolism. So both of these things are a great energy source, and proteins for sure for making enzyme, and for fats to make membrane, and as well as cell structure. So oh, you can mention all of these things here. So to provide nourishment or the nutrients which are required for further development, like fertilized egg, for the fertilized egg. And next, and with that, the proteins for making enzymes, as well as proteins. In fact, let me mention both of these, proteins and fats for making enzymes, cell structures, membrane structures, membranes, so on and so forth. With that, they are a great source of energy, for the metabolism, growth, division of the cell, and cell division. Okay, then describe the function of the jelly coat. Now, the jelly coat actually ensures that only one sperm fertilizes one egg, in a sense that jelly coat hardens when one sperm has fertilized the egg, and it would not allow any other sperm to enter the egg after the egg has been fertilized by one sperm. So, jelly coat hardens when one sperm has fertilized egg. Fertilize the egg, and prevents more than one sperm from entering the egg. Okay, then fertilization results in the formation of a zygote, and we need to describe how an embryo is formed when from a zygote. Now, after fertilization has taken place in the fallopian tubes, okay, the fertilized egg, like fertilization has taken place, the sperm has actually fertilized the egg. So later on, the zygote that has formed now, the zygote is going to divide, and it's going to continually divide till it moves down, the fallopian tubes and reaches the uterus. Okay, then then first the zygote is actually going to become like a solid ball of cells, and then it becomes like a hollow ball of cell, which is named as blastocyst. And inside the uterus, the blastocyst implants in the walls of the uterus, all right, where it's going to develop into an embryo, which would be attached to placenta and surrounded by fluid filled membrane. So after the fertilization, fertilization has taken place in the fallopian tube. So the um, zygote divides as it moves down the fallopian tubes to the uterus. Firstly, the zygote is going to divide into a ball of cells. Okay, firstly into a solid ball of cells, and then into a hollow ball of cell, which is named as blastocyst. And then this ball of cell is going to get blastocyst, or maybe you can say ball of cell, gets implanted in the walls of the uterus, where it develops into an embryo attached to placenta and fluid filled membranes. So these are all of the things that you need to mention, and let's talk about the next question. So the next question says that antibodies are proteins that are produced by lymphocytes. Now, remember that antibodies are like the soldiers of your body. If any foreign species or foreign object enters your body, foreign pathogen enters your body, what happens is that if your body has got the antibodies for that specific pathogen, antibodies are very specific for the type of pathogen. So, if your body has got the antibodies for that specific pathogens, so the specific soldiers are going to come out and fight with that pathogen. Now what now sometimes what it's like that our body is not really familiar with the specific pathogen. Then what happens is that our body won't really have those specific type of antibodies for that pathogen, or your body won't really be prepared for that specific type of viral infection or any infection like that, so because it does not have the solve for for that specific task. So what scientists do or what doctors do is that vaccination, you would have heard about it. So vaccination is a way of providing the active immunity. Now what is that? The dead pathogen vaccine is like a dead pathogen being entered into your body, so that your body gets familiar with it and produces the antibodies which are specific for that pathogen. The pathogen is not going to attack your body, but your body is going to get familiar with that pathogen, or the soldiers are going to get produced for that specific type of pathogen. Okay, so now your body is prepared against that specific disease, right? Now whenever the real pathogen enters your body is prepared for fighting through it, and you would survive that viral attack or any pathogen attack. So that's what a vaccine does. Now what is an antitoxin? Now antitoxin is going to neutralize the toxin. Now if antitoxins are kind of antitoxin antibodies, if they are provided to a patient, that is like a type of passive immunity, like a temporary. Active immunity is not a temporary solution, in fact it is long term solution. But when we talk about passive immunity, so it is a very temporary type of solution. um Passive immunity means protection to that person, like fast protection to that person from that disease until the body produces its own antibodies. Like your body was not, consider that there was a pathogen which your body was not really familiar with, or there was a toxin which your body was not really familiar with. So antitoxin is provided to your body. Antitoxin antibody is actually going to generate the passive immunity. That means a fast protection to that person from that disease would be provided until the body produces its own antibodies. So that's that. Okay, so explain the advantage of giving the person an injection of antitoxin. So introduction of antitoxin antibodies is going to generate passive immunity, so that would provide fast protection to that person from that disease until the body produces in its own antibodies. But it is a temporary solution. Okay, it is a kind of passive immunity, it is temporary. Okay, then explain how the two injections of the vaccine result in a better protection against the diphtheria than the injection of antitoxin antibody. So vaccine is dead pathogen, so by injecting the dead pathogen dead by injecting vaccine, okay, what you are doing is that into someone's body you are generating active immunity. You're generating active immunity. And antitoxin antibody is actually like a passive immunity. Active immunity is that means by exposure to the diseased organism, a disease causing organism, it triggers the immune system to produce antibodies for that disease. So your body would now produce the specific antibodies for that disease, and now your body would be able to, when the real pathogen enters your body would be able to fight through that disease. So that is active immunity, by exposure to pathogen. Because active immunity, because you're providing vaccine contain the death pathogen, so your body has got exposed to the pathogen, to the exposure to the pathogen triggers the immune system to produce antibodies against that pathogen. And when the original organism or the original pathogen would enter into the body, now your body is prepared for the for fighting against through that disease. So uh, and the other one, like the antitoxin was actually um, passive immunity. Passive immunity. So that's the difference. I hope you understand. Now explain how antibodies protect the body against pathogens. So okay, so it's like the pathogen acts like an antigen. Remember that if you have heard of lock and key model and for the enzyme, it is similar to that. Now pathogen acts like an antigen. The antibodies that are present inside your body, those are really specific for the type of antigen that gets attached to them. Okay, it's something like this. Okay, this is the antibody. This is your antibody, and it is very specific for the type of antigen that comes and gets attached to it. Antibody, antigen. Okay, so whenever a pathogen जो होता है, pathogen acts like an antigen, and the antibodies that are specific for that antigen, those are produced inside your body. ठीक है? And the antibodies are very specific for the type of antigen. So those antibodies are going to come and lock. They are going to the antigens are going to get attached to those antibodies, and then those antibodies are going to digest that pathogen, are going to destroy the pathogen. So that's the whole crux of it. Try writing more than like at least 4 points describing all of this activity. And yeah, so antibodies can travel through the body in the lymphatic system. Oh, just a minute. So we are going to talk about. Okay, so state two functions of the lymphatic system other than defense against the disease. So one of the functions is that your lymphatic system actually absorbs or maybe transports the fatty acids and glycerol. With that, it also drains the tissue fluids and returns the fluid or lymph to blood or the plasma. So absorbs, or you can say transports fatty acids and glycerol and glycerol. With that, it drains the tissue fluid, um, or you can say returns fluid to, or returns fluid to blood or plasma. All right, that's that. Let's just uh, do, do this a little bit in detail so that we can end this lecture. So what happens is that pathogens, we say that pathogens have antigens. In fact, not acts like. In fact, let's say pathogens have antigens. That would be a better term. So pathogens have antigens and antibodies lock onto. Okay. those antigens or the pathogens. Now, antibodies are complementary, are specific for that antigen. You can say case lock onto that antigen. So antibodies lock onto that antigen, and the antibodies are specific or we have got a complementary shape for that antigen. Okay, and where that way what happens is that the antibody is kind of going to mark the pathogen for destruction, to destroy the pathogen, and that destruction might happen by phagocytes. So phagocytes. Now phagocytes are play a great part in your immune system. Phagocytes work by ingesting the pathogen inside themselves. They are pathogen eaters. ठीक है, वो क्या करते हैं? They would ingest the pathogen in themselves and then destroy it. And yeah, now it's clear, I guess. And that would be it for this lecture. I hope you understood, and that would be it, I guess. Okay, so we have completed a June 2021 paper, biology. Okay, then Allah Hafiz.